Hello, dear viewers. My name is Shimmer, and I am from DOI.bio. Today, we will start our deep dive into the paper entitled Simulating 500 Million Years of Evolution with a Language Model. Today, we will just focus on the authors, abstract, and introduction. Please welcome my colleague, Echo, who will be helping today by reading the paper.
Thank you, Shimmer. My name is Echo, and I'm also from DOI.bio. Let's start with the authors of this paper, who are Thomas Hayes, Roshan Rao, Halil Akin, Nicholas James Sofroniou, Deniz Oktay, Zeming Lin, Robert Verkwil, Vincent Quaitran, Jonathan Deaton, Marius Wigert, Rohil Badkundri, Irham Shafkat, Jun Gong, Alexander Derry, Raul Santiago Molina, Neil Thomas, Yusuf Khan, Chetan Mishra, Carolyn Kim, Liam J. Barty, Patrick D. Shu, Tom Surku, Salvatore Candido, and Alexander Reeves.
The authors in this paper are primarily from Evolutionary Scale, an exciting new startup developing protein large language models. 3 are from the ARC Institute, and 2 of those are from the University of California at Berkeley. Alexander Reeves is a significant figure in the field of protein structure prediction, particularly known for his work at Meta AI, where he led the team responsible for developing ESMFold. ESMFold is a cutting-edge tool that uses large language models to predict the three-dimensional structure of proteins directly from their amino acid sequences. This approach leverages an earlier ESM model, ESM2, a massive language model trained on vast amounts of protein sequence data. Reeves was previously a doctoral student at New York University, working under the guidance of renowned AI researchers Jan LeCun and Rob Fergus.
Abstract. More than 3 billion years of evolution have produced an image of biology encoded into the space of natural proteins. Here we show that language models trained on tokens generated by evolution can act as evolutionary simulators to generate functional proteins that are far away from known proteins. We present ESM3, a frontier, multimodal, generative language model that reasons over the sequence, structure, and function of proteins. ESM3 can follow complex prompts combining its modalities and is highly responsive to biological alignment. We have prompted ESM3 to generate fluorescent proteins with a chain of thought. Among the generations that we synthesized, we found a bright fluorescent protein at far distance, 58% identity, from known fluorescent proteins. Similarly, distant natural fluorescent proteins are separated by over 500 million years of evolution.
This abstract describes a significant advancement in computational biochemistry and synthetic biology, specifically in the realm of protein engineering via deep learning models. The central focus is on ESM3, a cutting-edge generative language model tailored for protein engineering. The authors of this work have managed to leverage the ongoing advancements in AI, particularly in natural language processing, NLP, and apply it to the language of proteins, sequences of amino acids that, much like human languages, have evolved over billions of years. In biological terms, proteins are fundamental to life, with their diverse functions driven by the unique three-dimensional structures they fold into, determined by their amino acid sequences. Evolution has been the driving force in exploring the vast space of possible protein sequences, a process taking over 3 billion years. The ESM3 model seeks to emulate this evolutionary process by generating protein sequences that could feasibly occur in nature, but haven't been discovered or explored yet. The key achievements and characteristics of ESM3 highlighted in the passage can be summarized and elaborated as follows. 1. Multimodality. Unlike simpler models that might only consider the amino acid sequence of proteins, the primary structure, ESM3 is multimodal. That means it takes into account not just the sequence, but also the three-dimensional structure, tertiary structure, and functional attributes of proteins. This comprehensive approach allows it to more accurately predict how changes at the sequence level will impact a protein's overall structure and function. 2. Generative capability. ESM3 is not merely predictive, but generative. It can create novel protein sequences that are significantly different from those already known. This is a marked departure from models that can only analyze existing sequences. ESM3 pushes the boundary by proposing new sequences that, while novel, are plausible within the biological rules governing protein structure and function. 3. High responsiveness to biological alignment. The model's design allows it to respond effectively to prompts that require consideration of evolutionary relationships and conservation. Its ability to factor in biological alignment means it can appreciate the significance of sequence identity and evolutionary distance in its generation process. And 4. Application to fluorescent protein design. The authors put ESM3 to the test by using it to design fluorescent proteins, a class of proteins commonly used as markers in biological research. They successfully generated a new fluorescent protein that shares only 58% sequence identity with known variants, suggesting a significant evolutionary distance equivalent to over 500 million years. This accomplishment underscores the model's power not just to iterate on known proteins, but to leapfrog across vast evolutionary distances to find entirely new variants. 5. Chain of thought prompting. ESM3's ability to follow complex prompts, possibly including a series of logical steps or conditions akin to a chain of thought, indicates a level of interaction sophistication that can mimic human reasoning to a certain extent. This feature likely enhances its generality and flexibility in generating functionally targeted proteins. The implications of this work are profound, touching on both basic biological research, where it could greatly accelerate the discovery of new proteins with functions that haven't been seen before, and applied fields like drug design, where the ability to custom design proteins with specific structural and functional properties could revolutionize the development of new therapeutics. It represents a remarkable convergence of computational science and molecular biology, with the potential to unlock a new era of synthetic biology and protein design.
We continue with reading the paper. Introduction. The proteins that exist today have developed into their present forms over the course of billions of years of natural evolution, passing through a vast evolutionary sieve. In parallel experiments conducted over geological time, nature creates random mutations and applies selection, filtering proteins by their myriad sequences, structures, and functions. As a result, the patterns in the proteins we observe reflect the action of the deep hidden variables of the biology that have shaped their evolution across time. Gene sequencing surveys of Earth's natural diversity are cataloging the sequences.
To support this, the authors use the following references. Reference 1. UniProt Consortium. The UniProt database serves as a comprehensive resource for protein information, aggregating data about protein sequences, functions, and structures. It plays a crucial role in bioinformatics and biological research by providing standardized annotations and a user-friendly platform to explore protein entries, enriching our understanding of protein biology and functionality. Reference 2. Igor Grigoriev et al. The Genome Portal of the Department of Energy's Joint Genome Institute offers access to genomic data, enabling researchers to explore a vast array of sequenced organisms. The portal is invaluable for studying various genomic features, facilitating advancements in fields like ecology, evolutionary biology, and bioenergy, ultimately helping us understand the genetic diversity of life on Earth. Reference 3. Alex L. Mitchell et al. mGenify is a microbiome analysis resource that provides tools for the characterization of microbial communities. This resource includes data on various microbiomes, allowing researchers to investigate microbial diversity, functions, and interactions, thus enhancing our understanding of the critical roles that microbes play in ecosystems and human health.
We continue where we left off. Gene sequencing surveys of Earth's natural diversity are cataloging the sequences and structures of proteins, references 4 and 5.
The reference used for the structures of proteins are reference 4. Mihaly Varady et al., the AlphaFold Protein Structure Database, launching in 2024, will house structural information for over 214 million protein sequences, a milestone achieved through advanced computational methods. This extensive database aims to support research by helping scientists predict protein structures, offering insights into their functions and interactions in biological systems. Reference 5. Zemming-Lin et al. This study introduces a method for predicting atomic-level protein structures using a language model. By leveraging evolutionary information and deep learning techniques, the researchers have demonstrated that language models can outperform traditional methods for protein structure prediction, paving the way for innovative approaches in protein engineering and design.
containing billions of sequences and hundreds of millions of structures that illuminate patterns of variation across life. A consensus is building that underlying these sequences is a fundamental language of protein biology that can be understood using large language models, references 6 to 10.
Reference 6. Ethan C. Alley et al. This research emphasizes a unified approach to protein engineering using deep learning techniques. By transforming sequences into a comprehensive representation, the study highlights that intelligent analysis of protein sequences can lead to significant advancements in the design of new proteins with targeted functions, streamlining the protein engineering process. Reference 7. Alexander Reeves et al. This groundbreaking paper shows how unsupervised learning can extract biological structure and function from over 250 million protein sequences. The results indicate that scaling up training data allows for emergent capabilities in understanding protein properties, which can lead to enhanced algorithms for predicting protein behavior and functions. Reference 8. Ali Madani et al. This study demonstrates that large language models can generate functional protein sequences across diverse families. By training these models on extensive datasets, the researchers succeeded in producing new protein sequences that can potentially exhibit desired biological activities, highlighting the power of computational methods in protein discovery and design. Reference 9. Noelia Ferouz et al. ProtGPT-2 is an innovative deep learning model designed for protein design. By using an unsupervised learning approach, ProtGPT-2 can generate novel protein sequences that may have practical applications in biotechnology and pharmaceuticals, emphasizing the potential of artificial intelligence in advancing protein engineering. Reference 10. Robert Verkuil et al. In this study, the researchers explore how language models can generalize beyond natural protein sequences, showcasing their ability to understand and predict protein structures. The findings reinforce the idea that these models are not only effective for existing proteins, but can innovate new sequences that could serve various biological functions.
A number of language models of protein sequences have now been developed and evaluated.
The references here are Reference 9. ProtGPT-2, a revolution in protein design. In this landmark study, published in Nature Communications, Noelia Ferouz and her team introduced ProtGPT-2, a groundbreaking deep, unsupervised language model specifically tailored for protein design. By utilizing advanced machine learning techniques, ProtGPT-2 can generate protein sequences that could lead to innovative biotechnological applications. This research opens up exciting possibilities in synthetic biology and medicine. Reference 11. ProtTrans, Cracking the Code of Life. In a mesmerizing exploration titled ProtTrans, Ahmed El Nagar and colleagues dive into the realm of self-supervised deep learning to decode life's fundamental language, protein sequences. Published in the IEEE Transactions on Pattern Analysis and Machine Intelligence, this paper emphasizes high-performance computing to enhance protein understanding. ProtTrans empowers researchers by providing powerful tools to analyze biological data, paving the way for breakthroughs in bioinformatics and computational biology. Reference 12. RITA, Scaling Up Generative Protein Models. In July 2022, Daniel Heslow and a diverse team published a compelling study titled RITA, which focuses on scaling up generative models for protein sequences. This research showcases innovative methods to enhance the capability and efficacy of existing models, ultimately refining how scientists can generate and evaluate protein structures. RITA stands at the forefront of computational biology, providing insights that could revolutionize protein engineering. Reference 14. Evolutionary diffusion for protein generation. In an exciting 2023 preprint on Bioarxiv, Sarah Alamdari and her team propose a new approach for protein generation using evolutionary diffusion. Their innovative model demonstrates that sequence data alone can yield impressive results in generating functional proteins. This study challenges conventional wisdom in protein design by highlighting the power of simple sequences.
It has been found that the representations that emerge within language models reflect the biological structure and function of proteins, references 615 and 16.
First up, in reference 6, we have a study by Alley et al., published in Nature Methods. In their 2019 paper, the the researchers introduced a unified approach to protein engineering using sequence-based deep representation learning. The key innovation here is the integration of various data types and methodologies for optimizing protein sequences. By employing deep learning models, they achieved remarkable accuracy in predicting how changes in protein sequences could influence their functions. This research highlights the potential of AI in streamlining the protein engineering process, making it quicker and more efficient. Next, let's discuss reference 15, a study by Heinzinger and colleagues published in BMC Bioinformatics. In their 2019 work, they explored the idea of transfer learning applied to protein sequences. They modeled aspects of the language of life by using deep learning techniques to capture the complexities of protein structures. Their findings showcase how transfer learning can drastically improve our ability to interpret biological data, facilitating better understanding and predictions regarding protein functions. This work is a critical step towards enhancing computational tools in bioinformatics. Finally, we look at reference 16, a 2021 study by Meyer et al., which pushes the boundaries even further. They showcased how language models can enable zero-shot predictions regarding the effects of mutations on protein function. This means that, without prior training on specific data, their model could still effectively predict how mutations would impact proteins. Such capabilities could revolutionize how scientists rapidly assess and modify proteins, particularly in the fields of drug design and synthetic biology.
And are learned without any supervision on those properties, improving with scale, references 5, 17, and 18.
The references presented here are Reference 5. Zemming-Lin et al., 2023. The study presents a model capable of predicting atomic-level protein structures by leveraging a language model that has been trained at an evolutionary scale. This work highlights the ability of unsupervised learning approaches to effectively capture complex properties of protein structures from large datasets. Reference 17. Roshan Rao et al., 2021. This paper discusses how transformer-based protein language models function as unsupervised structure learners, indicating that these models can discern structural properties of proteins without the need for explicit supervision. The authors underscore the model's capability to learn from data at scale, thereby improving their performance on structure prediction tasks. Reference 18. Bo Chen et al., 2023, the authors introduce Extremope-BLEM, a large-scale pre-trained transformer model designed to interpret the language of proteins.